Hi friends, this is Bhavna Chaloria and today I am going to discuss about the topic named as DNA origami tiles at nanoscale mazes. The very first question you should know that is what is DNA origami? The DNA origami is a technique in which nanostructures are created into shapes by folding stranded DNA with staple strands. So this is the picture of origami which is created by staples plus template. Nucleotide bases are adenine, guanine, thymine, cytosine. They form complementary bonds which are AT or CG. These DNA sequences should be in the right order that produces shapes. So here's the introduction. Nanostructures are have been used as scaffolds to organize diverse molecules such as dyes, proteins, metal nanoparticles, and polymers. Because of this spatial precision and high programmability of DNA structures, researchers said that the functional component into molecular devices and for advancing a variety of technologies including plasmonics, molecular electronics, and photonics. Why do we need DNA tilings? We need DNA tilings for the complex networks. Stochastic algorithms provide solutions to create complex networks. So, the random yet controlled property is referred as a programmable disorder. By using DNA self-assembly, program disorder can now be generated. Here is the one example of a rhodopsin photopigment expression in the flying retina. Here you can see that the green fluoring receptors are 70% and blue fluoring receptors are 30%. So this process is totally random. So the ratio of blue to green in photoreceptor cells indicates that the choice is not simply stochastic. So these processes are totally random. Here's another example. DNA origami nanostructures. You can see that blue and gray with internal patterns, red, used as a building blocks to generate program disorder. Here you can see that biological networks has now been demonstrated with DNA origami tiles using a stochastic algorithm. There are two studies for constructing large-scale arrays of DNA tiles. First is periodic arrays and second is algorithm arrays. Origami tiles are large enough to accommodate complex internal patterns for organizing molecules. There are three designs of scaffold folding paths for square DNA origami tiles that is symmetric and allows stacking bonds on all four sides. First page shows that a scaffold path that fills in four isosceles right triangles sequentially. Second pick a scaffold path that fills in four square shaped quadrants sequentially. Third pick a scaffold path that fills in four isosceles right triangles sequentially. So, to scale up the complexity and diversity of origami arrays, we present a strategy using random DNA tilings. The strategy is stochastic trajectory tiling. Now, what is trajectory tile? A trajectory tile has a rotationally asymmetric pattern that is designed to continue into neighboring tiles. So here's the trajectory tile. You can see that the trajectory tile is split along the diagonal into two triangles of contrasting colors, and so that the four possible orientations has been shown. Here are some examples of surface filling made tiling such a pattern. With this scheme, you can see the pictures with random placement. Here's the quarter circle of a tile, the texture tile. If you rotate this tile at a 90 degree, then you will be arrived with this pick. So the DNA origami nanostructures as algorithm nanoscale building blocks to utilize structure tiling. Here above diagrams are the representative patterns that contain program disorder. One is loop, second is maze, third is tree. So in maze, varying internal patterns on the tiles created maze-like patterns with different branching rules. In trees, programming the matching rules between the tiles allowed the creation of trees with different growing directions. And in the last one, the is loops. Adjusting the ratio of different types of tiles gave trees and loops with controlled dimensions. Here's the summary of a programmable disorder in random DNA tilings. Program tile. Here you can see that we have programmed this programmable tile in the software where xi or of yi defines the start and endpoints of a line and cxi cy defines the center of an arc. Here's another example of a program grid in programmed in the software, defining a grid. Here, I, J indicates the location on the grid. Oriented defines the orientation of a tile. As the last one, program random choice. Here, oriented defines an orientation of the tile and pi is the probability of the orientate. So these are the mazes, trees, and random choice. So the sizes of loops, mazes, and trees are defined on the basis of the area that they cover. The creation of a maze-like nanostructures with programmable disorder demonstrates the power of stochastic algorithm self-assembly. A stochastic algorithm provides a simple solution for producing a wide variety of sophisticated arrays. So here is the conclusions. Developed a framework for creating programmable disorder in random DNA tiling, created DNA nanostructures with a diversity of complex patterns including random loops, mazes, and trees. Stochastic algorithm provides a simple solution for producing a wide variety of sophisticated arrays in a massively parallel fashion. Mazes made of a DNA origami nanostructures can provide a wide range of test conditions for DNA mortars or walkers. The massively parallel production of complex nano-objects with programmable disorder could be combined with inorganic or biological functions materials to evolve molecular devices with novel functionalities that rival the biomolecular machineries in living systems. Here's the references. Thank you.